So basic differential expression analysis is, the title for this, but a lot of the concepts can apply for their omic analysis, right? So what's the difference between omic analyses and non-omic analysis is that, traditionally we focus on one molecule and we do the testing for that. But omic allows you to test a set, the entire transcriptome, the entire proteome, et cetera. And it comes with its own sort of considerations. Okay, so by the end of this lecture, you will understand the key steps in identifying differentially expressed genes using RNA-seq, bulk RNA-seq, which is an assay to measure transcript levels. And it's a sequencing-based assay. It's not a microarray-based assay, okay? Which means that the resulting data have different properties. So you're not going to take a library that's designed for microarray RNA-seq and RNA analysis and apply it here. And now we're going to introduce you to considerations for multiple testing because, as Spider-Man says, with great freedom comes great responsibility, right? So now, his uncle, thank you. So now you have this amazing ability to measure all the things in the cell, right? All the genes. Now, if you were to do multiple testing on all of those genes, you are going to set yourself up to find a lot of false positives. So how do you tell how much signal is in your data set? How do you correct for false positives? And why do you need to correct for them, right? Those are the kind of topics we're going to cover now. We're going to introduce you to ideas such as looking at a histogram of your p-values, because now you don't have just one p-value for one gene, you have 20,000 p-values for 20,000 genes. And so you got to look across the whole thing and say, which of this is real signal, right? And there are some methods to do that. And then we're going to look at some very simple visualization tools to look at the result of this high throughput differential gene expression analysis. So whenever you got a new type of assay you're studying, I recommend you go look at a review article or a YouTube video or whatever on what the steps are to generate the data you're looking at. So if you're doing single cell, what kind of single cell assay is it? How are the data generated? It's very important to know where the data come from, because that will remind you about the sort of QC checks you need to make along the way. And it might alert you to other things that might be missed in somebody else's pipeline if you've gotten it. So it's always good to know for yourself. Okay. So, here we show 3 different assays used to measure RNA with sequencing technologies. They all have related workflows here, and I'm briefly going to go over the workflow. So let's start with your classic short read sequencing, which is when people say RNA-seq, that's usually what they're talking about. So how does short read sequencing work? You start with your RNA, you fragment it, your gene can be like kilobases long. Your reads, when they come off a sequencer, are not kilobases long. They're like 150 base pairs or whatever, right? So what part of the RNA are they measuring? So here you're going to get the RNA. It's going to be fragmented to accommodate the limitation of the sequencing technology. And because RNA is fragile, you are going to convert it to cDNA, which is complementary DNA. Then you are going to add some sequencing adapters onto it in order for that molecule to travel through the particular sample prep of the technology, right, that you are using to generate your data. So if you are using an Illumina sequencer to measure your RNA, Illumina is going to give you a kit. It's going to have little adapters, and the adapters are going to go onto your reads, right? Onto your molecules. And therefore, when the sequencer sequences them, it's going to sequence the adapters as well, right? And the adapters have various purposes. They're used to tether your target molecule to whatever substrate in the flow cell. the sequencing occurs with, because once you've got this adapter ligation, you're going to take this molecule and then you're going to amplify the signal so that you can measure it, right? So in your actual cell, you have very few copies of the gene, but now in a lot of assays, you amplify it by PCR internally, right? So what does that do? It introduces biases as well, because some sequences PCR amplify better than others, right? So the other reason you need to know why, where your data come from is so that you can then learn to expect what you would see if you visualize the data at a particular level. So if I got just my fragments, right? I'm jumping ahead of myself, but basically when you visualize your fragments, they should look a certain way. They should be spread out across the gene. They shouldn't just all be at the 3' end, for example, right? PCR bias. Oh, does PCR sometimes, over duplicate a molecule? And do I know I need to deduplicate it? Yes, you do, right? So these are things that you become aware of. And so what I suggest is with every workflow, you have a certain number of steps for sample prep, and you systematically ask yourself, step one, what biases can be introduced? Step two, what biases can be introduced and how do we correct for it? And so forth. And then make sure your QC pipeline has that, right? Adapter ligation, G. If you sequence something that has adapter ligated, you better have some way of informatically taking those adapters off later. And yes, you do, right? And which adapter, what's the adapter sequence? Well, it depends. Which chemistry of alumina are you using, right? So these are some things that you will be more aware of if you know what the process is. And then finally, there's for short read sequencing, there's some kind of size selection, again, to deal with the limitation of the sequencing technology. And then you get what's called a read. Okay, so what is a read, informatically speaking? What does a raw data look like for RNA-seq? So if you got a read for RNA-seq, it just came off the sequencer, right? People say this, it came off the sequencer. So what do you get? That's right. It'll be a bunch of strings, right? That's the term for it. It's like strings. Um, and so then, yes, adapters are different than primers.
How do the adapters affect like the parameters? So they will be like go on the end of the fragment.
I think of yeah. So I'm not 100% sure, to be honest, but my understanding is that a primer is used for extension, and sometimes the adapter is like a primer has an overhang, whereas an adapter doesn't. That's kind of how I think about it, right? But I think a primer is a more general term for any kind of oligonucleotide that's used to cause synthesis of the complementary strand. So maybe adapters are a particular kind of primer, right? But
So if you're using adapters, you still use primers as well.
maybe the primers are for the adapters. Yeah, I have to be honest. I don't know exactly what the difference is between the two, but my understanding is that primer is just a general term for, yeah, I need to amplify the sequence, so I need to give enough of a complementary a nucleotide sequence in order to uniquely identify that sequence and start amplifying it. Whereas adapter is usually in the context of a sequencing workflow, right? So if you've got single cell data, you're going to have adapters added, which indicate which cell the sequence came from. Um, and then there are other adapters too, which are needed in order to get this molecule to connect to the flow cell and be sequenced. So adapter is very much in the context of the technology, and primer is just general as far as I know. Yeah. Okay. And the other thing you should know is that, there's many flavors of RNA sequencing. You've got short read sequencing, you've got long read sequencing where the reads can be kilobases long, and there's technology such as the Pacific Biosciences technology. And then you've got, direct sequencing, which does not involve PCR. So nanostring is an example of direct sequencing where you don't do PCR amplification. You sequence exactly the molecule you got, right? So the sensitivity isn't great, but it doesn't have the biases of PCR. Okay. And so the thing you have to remember is that sequencing RNA is not the same as sequencing DNA, because RNA, if you've got a particular gene, you can have multiple transcripts of the gene. And one of the problems in RNA-seq is transcript quantification. So you have to see which parts of the nucleotide sequence belong to transcript 1 versus transcript 2, and vice versa. And so you have to have, when you have a gene expression quantifier, it's got to be transcript aware. It's got to know that, this gene has that many transcripts, and you get that information from a gene database. The other thing is, the other thing you have to be aware of is what kind of RNA are you getting? Are you getting just poly RNA, or are you getting everything, right? Are you getting just messenger RNA, or are you getting ribosomal RNA, non-coding RNA, tRNAs, et cetera, et cetera? There's like a zoo, right? So accordingly, when you quantify your gene counts, your aligner had better be compatible. It better have the right set of gene definitions. If you only gave it a definitions comprising of mRNA, et cetera, and your assay is picking up on non-coding RNAs, those are going to be missed, right? For example, the other thing is your process has to be aware of the phenomenon of splicing, right? So in the genome, you have two exons here. They may be separated by a very long intron, right? And there's going to be, so when you have a read from a piece of spliced RNA, those two exons are going to be joined, right? And then the algorithm that goes to align that piece of sequence, it had better know that these two parts of the genome can either be far apart or they can be connected. Otherwise, it's going to throw out anything that's been spliced, right? This is another example of pick the right tool for your application. So if I'm doing DNA sequence alignment, I don't have to worry about being splice aware. But if I'm doing RNA sequence alignment, I do. Okay? So the whole point of this is to say, even RNA-seq comes in many flavors, many read lengths, and just pick the pipeline that's right for your needs and be aware that there's different considerations. Okay. So now we have gone through that process and we have generated data. Okay. So as you said, the raw reads that come off the sequencer are a set of strings. They're just letters. We don't know where in the what in the genome they correspond to. Somehow you have to put it through a pipeline, and at the other end, you get a nice table of gene expression by samples, right? So what's happening? Thankfully for all of us, the informatics community has agreed on a set of standards to represent these data types and represent them in particular file formats that speak to the different stages of the processing. So the field, agrees that the reads that come off the sequencer should be, represented in a FASTQ format, okay? What is a format? You can just go look it up, right? So the point is, informatics, we love our file formats. We are awash in file formats. For any given file format, there is an R package for you to read it and convert it to some other kind of file format. If it is a common enough thing to do, somebody's written the code to do it. Do not do it from scratch. So would I sit and open up a FASTQ in a text editor? I could. It's human readable, right? You can do a head command on a FASTQ file, and you will see its file format. But I don't need to. I can just throw it into the pipe. That's good to know. Okay, so you get your raw reads. Then what you do is you take your reads and then you have to align them to the genome, right? So this is like a computer science problem, is like, given a string of a certain length, uh, where in this longer string does it align to, right? And so in a lot of times, like I'm telling you this, but if you generate this data, especially from a genomic score, I would say, see if for a nominal amount of, a nominal charge, they'll run it through their pipeline for you and give you the gene count matrix. Because if you're not doing this all the time, there's no point in you trying to set up the aligner. And an aligner needs to be needs to have index files for whatever genome you're aligning it to. So if I'm a lab that does mouse work and you're a lab that does human work, I'm probably going to have the indexes set up for mouse. And if it's a very common aligner tool, you can probably download the human index from somewhere on the internet, right? For the right build of human genome 38 or whatever. Like, and you probably, I don't know what you folks do in metagenomics. Yeast cell, yeah. But the bottom line is that, practically speaking, um, there's this workflow. It starts with reads, no matter what. Even if you get single cell data, it's just reads. And at some point, you need to align it to the genome, and the aligner has to be aware of it has to be the right aligner for the data type you're using. So for RNA-seq, it's got to be splice aware, right? For DNA, it need not be. For methylation data, the methylation assays sometimes use techniques that convert the bases, right? They convert the C's, some C's to T's, and they leave other C's as is. So the aligner has to be methylation aware. You can't just toss it into a regular aligner. It won't align well. The other thing that happens is once you align reads to the genome, there are going to be some reads that map uniquely and some that map in a few different places. And usually what we do is we throw out the ones that map all over the place. There, and then that happens to be its own subfield. People look at that like, oh my God, there's a duplicated gene family that's evolved, and it's involved in human brain expansion, but nobody studied these genes because whenever the aligner aligns the reads, we throw them out. Like, that's fine. But to simplify, start simple. Uniquely mapping reads, okay? Then you have you have something called a BAM file or a SAM file. That is basically a way of, it encodes where all of the strings map to in the genome, and each alignment is its own record in the file, and it'll tell you how well it aligned, how many mismatches there were. Like, every alignment gets a score based on whether it's a perfect match or there was a slight mismatch. Maybe mismatches are okay because there's variation in the population, right? You guys know what a SNP is? A SNP is a location in the genome that varies in 1% of the population, or 5% of the population or more, right? That's the major source of human variation. Like, our DNA is not like 100% identical. There are hotspots in the genome where there is a high degree of population variation, right? So if the aligner was always looking for perfect matches, it would throw out a lot of these reads, right? So these are the kind of things. So the aligner keeps score, and then finally, it bins things. So now we have mapped the reads to particular pieces of the genome. Now it's time to count how many reads fall into this exon, how many fall into this transcript, and you bucket things like, okay, this read maps to this exon, and this exon is only in this transcript. Therefore, this read maps to this transcript. All that is done, and then you get a nice table on a platter, right? Finally. So you get your transcript counts. Then you have to do some kind of normalization or equalization for variation in sequencing, right? So you put in 2 samples into the sequencer, there might be some variation just because you happen to sequence a bit more of this than that one. Doesn't necessarily, if you didn't normalize, it would look like the genes are up in here compared to there, but they're not. It's just that you happen to sequence more for the sample, right? It's just because there's stochasticity in this process and sampling variation. So you need to control for that. So that's what normalization does. And then finally, we now can take the genes and we can say what's different between A and B. Oh, so there's stochasticity in how the sampling occurs, right? But then you normalize for it by dividing by the size of the library. So you just see that this sample has twice as many reads overall, and you just normalize it so that every library size is like 100 reads. Yeah, that's right. That's right. It is within your own data set, but it's from sample to sample. Yeah. So if I took the same person's RNA and I sequence it 3 different times, you'll have something called the library size, which is the total number of reads you got from that person or counts you got for that person. The same person is going to have variation just because of the way the nucleotides flowed and got sequenced and PCR and all that stuff. And so you just sort of normalize for that. Yeah. Callisto. I do not know much about Callisto. Why are you asking? Mm-hmm.
But because I was given data that's previously been aligned, so I don't
Okay.
know much about that back end process. But I think like Callisto, it sounds very familiar, so I think it like does those steps, I guess. But is it like something that's fully automatic, or if you're not familiar with it, sometimes
Well,
there, I was like, oh, I know that word.
yeah. So it's probably just a step in the pipeline, and it's just probably a splice-aware aligner. I do not know what the difference is between Callisto and Salmon, and for that, you'd probably have to look at a review article that's comparing different algorithms. But one thing you can do, for example, is you can look at a slide like this and say, okay, they got it to step 2, and now I have to do the downstream steps, right? So from a practical perspective, you just need to know that you just ask them, did you use standard parameters when you aligned it? Or did you change something? Did you tweak something, right? Did you make it very sensitive to alignment? Or did you just use the default parameters, right? And then you might want to consider if they change the default parameters, why did they? Right? Does it have to do with your application? Like, oh, for our data, we have to change the parameters for XYZ. It necessitated it, right? Those two things. But from the perspective, I mean, I haven't worked specifically with Callisto, but it sounds like just a splice aware aligner. Yeah. And a transcript quantifier.
I've just sent you a link to our RNA-seq sheets, like information on Callisto specifically. Full site's great. I like that one.
Thank you.
No problem.
Great.
I was gonna ask, follow up on the question about stochasticity, and you're talking about, like, in particular, patient sample, you're looking at the library from the same ones, but what do you do if you have, like, say, a postmortem rat brain at different embryo stages, and you can't compare that same sample again, because it's all been put in one collection? Or do you, if you wanted to get the entire thing, how do you handle stochasticity in that situation?
That's a great question. This is why when you are merging across data sets, you start, you make sure that, so what informatics people like to do is they just analyze everything from scratch, because then you know exactly what was done to the data, right? But if you don't have the bandwidth for that, And suppose you've got your older rat brains and your younger rat brains, and you're getting the older rat brains from some data set in a publication. You read their methods, and in their methods, they should tell you, they should give you enough details so that you know what was done. If they say, oh, we aligned it using, XYZ, with the default parameters, maybe make sure that your young rat brains also are aligned with the same thing, right? So they're all treated the same way. But then what you do is you try to kind of go back to, obviously you don't want to do extra work, right? So you just make sure you go back as far as you need to so that you can start analyzing them together so you can merge them, right?
Short follow-up to that then. So
Yeah.
It's like, I know, I haven't really done anything with RNA-seq yet, but I've done a lot of stuff with like Western blot, and like Western blot, oftentimes you're taking like a housekeeping gene for a load control.
Yeah.
And you're talking about how like different, with RNA-seq, different sequences might get amplified differently just because of the inherent variation in like in an RNA-seq. Can you still do like, take a housekeeping gene that you're not expecting much change at these like different developmental stages or these different conditions that you're looking at? Can you do something like that as well?
You wouldn't do that in this particular case. So for example, because the source of the bias is different. So if you are looking at a PCR amplification bias, then you have to have somewhere in your downstream model, when you're fitting the quantities of the gene, something like %GC in the model, if the field agrees it's big enough a problem that you would expect that. But normally, this pipeline doesn't show it in detail, but after assembly, usually sequencing pipelines have a PCR deduplication step. They remove duplicate molecules, okay? But then for other things, you kind of, you might sort of, if you have a genome-wide effect, you might stratify by %GC and say, this is a genome-wide effect, and whether if I stratify by GC, the effect still persists. You try to match sequences from somewhere else to control for that confound, okay? The normalization I'm talking about here, so for example, in your case, right, you mentioned getting the housekeeping gene as a loading control. Their variation comes from how much you're loading in your pipette, right? Yeah, so you're normalizing to that. Here, the variation comes from sampling differences for sequencing. So you normalize to like the total number of reads in that. The assumption is there's no inherent reason why systematically the cell should have across the board more transcripts than that.
You have like see, and maybe it's more difficult, right? Because high GC, it's more difficult to actually amplify that, so it might be a bias coming from.
It could be, but if you are comparing across samples, everybody's got that bias, right? So if you got a high GC gene, you got two genes, a high GC gene and a low GC gene, you're comparing the high GC genes across your cases and your controls, everybody's got that bias. You don't have to worry about that, right? But if you don't normalize for library size and your cases happen to have more, just on average, just by chance, then you might spuriously be calling upregulated genes in your cases when it's due to sequencing variation. Okay, so the solution must fit exactly the problem, right? So like we're scientists, so you want to, you have a confounding factor and you find a way to control for it appropriately, right? So and then at the end of it, and then at the end of this, you do the differential expression analysis. So what does a workflow look like in R? Let's assume that we start with raw expression count matrix, right? It's the output of tools such as STAR or RSEM. So what we do is, because we are doing multiple testing correction, one thing we'll talk about is that when you test lots of things, a fraction of them are going to be significant just by chance, just because of the nature of the statistical framework we use to call significance. So what you don't want to do is have a lot of uninformative stuff in there, which is going to serve no purpose other than to increase your false positives, So for example, if I have cases and controls and half the genes are identical across cases and controls, I might as well not test them, right? So you use some kind of, or half the genes are not even expressed. Why bother testing a bunch of zeros or near zeros? Because all that's going to do is it's going to make me test twice as many genes and increase my false positive by, twice. So 100%, right? So for that reason, a lot of omic assays, they start with this, then they will, remove genes that are like so low, we might not even bother. And it also takes up compute, so just remove those things because we know there's nothing happening there. We're looking for the big effects, right? Then we normalize for technical variation, and then we build our statistical model, which is going to be suitable for the kind of data we're working with. And then we run multiple hypothesis tests. So if you're doing RNA-seq analysis, you are in parallel doing hypothesis tests for each gene. That's why at the end of the day, a fraction of them happen to be significant, right? And those are your differentially expressed genes, and they pass some cutoff. And then you do the fun stuff. So for example, if you take protein coding genes and you just assume genes and not transcripts, you say, okay, maybe 20,000 protein coding genes, right? So you're going to have 20,000 statistical tests, 20,000 p-values, and the multiple testing burden. So you reduce the multiple testing burden, you control for variation in sequencing depth. Then you run this hypothesis test, and this is basically, you test two competing models. You say the expression of the gene can be explained just by the intercept, or the expression of the gene can be explained by intercept plus disease plus epsilon.
Okay.
Sorry?
Like, this is like B versus NX plus B, right? But M is disease, like,
No, M is beta 1. Sorry. You can just stick to the beta notations. MX plus B is just my high school math talk. That's how I think about it. It's like MX plus B. Yeah, but the betas, I think that in R, if you look at the output, they call them the coefficients, but you see this beta notation a lot. Yeah. And the algorithm fits both of these models, and it tries to see which one the data fit better, right? And if the data significantly fit the full model better, then those are your significant genes, okay? Okay. All right, that's your signal. And again, you did multiple tests. You pruned them at the top, but you're still doing multiple tests, so you've got to correct for it. And then finally, you look at the signal. All right, now we are going to stretch our fingers, and then we are going to do a worked example for RNA-seq analysis.